So following up on endocrine, it's Alice with the drink me bottle saying why it must be somatotropin, the growth hormone. Somatotropin, Latin for body responding. Those 2 years of Latin I took in middle school actually turned out to be useful. There's a certain lag in hormonal control. Quicker control systems involve nervous systems. Nerves are long tracts, extensions of cells. But somewhere along the cell is a support system, a cell body. It's off to the side of where the impulse goes, but it keeps the nerve going, right? Your nerves were put in place when you were an embryo, and they're still there. Is that you don't build new nerves, or you almost never build new nerves. You're using the ones that you've had your whole life. That's very typical for any kind of animal system. So you need a cell body to kind of keep it going. The tract, ideally, they're kind of like railroad tracks in that you can make an impulse, a nerve impulse, go either way on them. But they're set up in most animal systems so that there's a directionality to them. So the impulse is coming in along the dendrite, it's going out along the axons. Now, because there are animals where they'll use the same tract in both directions, for a while they were trying to discard the dendrite terms and just call them all axons and have directionality on them. But that does not seem to have succeeded as a change in terminology. But you may run into it some places. The systems typically work in a loop that isn't really a loop. You'll see why I put the word loop in quotation marks. How does the nervous system get information? It can't interact directly with the environment. It needs sense receptors to take environmental information and convert it into nerve information. So you have sense receptors that can pick up vibrations, that pick up pressure, that pick up light intensity, that pick up light movement, that pick up light colors, that pick up vibrations in the air, that pick up all sorts of different information and convert that into impulses that then go into the processing. Neurons, technically, that's what a nerve cell is. The neurons that carry information into the system are called afferent neurons. They used to be called sensory neurons, so you'll see that term in older sources. They connect to the central part of the system, the neurons that direct the flow, the neurons that interpret the information, the neurons that check against stored information, against memories, the neurons that think through what the response should be, the neurons that determine the response, the neurons that coordinate the response. So the central nervous system is full of interneurons. Something as simple as your spinal cord, which is a relay system, and as complex as the memory centers of your brain, which are storing information from your whole life, to the decision-making centers of your brain, to the center that once you've decided what you're going to do, coordinates how to send the messages out to the rest of your body to get that response to happen. That goes out along efferent neurons that used to be called motor neurons. And they interact with your muscles, right? Your response is going to be muscular. And they interact with certain glands, right? Oh my God, we need to run like hell. Part of that message goes to your adrenal glands and causes a release of adrenaline. And there's other hormonal responses that sometimes is coordinated out of the central system. So sensory receptors into afferent neurons, into inner neurons, out by efferent neurons to, now these are the one thing that isn't really nervous, is that your muscle and your gland system are responsives. Now a nerve is a different thing than a neuron. A nerve is a bundle of axons wrapped in connective tissue to keep them largely separate. Some axons are really large. In some animals, the size of the axon determines the speed that the impulse travels along the axon at. I'll get to that in a second. There's a whole bunch of cells called neuroglial cells. Your central system has its own support. And your neuroglial cells do a whole bunch of things that, okay, this gets complicated. Keep in mind, you have all of the genes for all the stuff that anything in your body needs to do. There are cells in your brain that activate genes, and they're essentially immune system cells. Is that there are communication cells, there are virtually hormonal cells. Remember, there's a big overlap between neuro and secretory anyway. Also, there are neuroglial cells that act in a lot of ways like connective tissue, and that they wrap around the individual axons in vertebrates, not in invertebrates. So there are neuroglial cells in all animals, but in invertebrates, these include what are called Schwann cells. Schwann cells, if you've ever seen somebody wrap tape around a wire to insulate the wire, where you stick it on there and then you go round and round and round and round and round. That's what a Schwann cell looks like, where it's a very flat cell, but it goes round and round and round around the axon, but it isn't actually stuck to the axon like the tape would be on the wire. There's a little bitty gap there. And what's in those cells is a protein called myelin, which is a pretty decent barrier to keep. You'll find out why in a couple of minutes. If you have either muscular dystrophy or multiple sclerosis, maybe both, then your immune system decides it doesn't like myelin. And it starts to go after these cells and strip them off of the axons. And your impulses kind of bleed away instead of going down the wiring the way that they're supposed to. And, that's pretty bad. Now, the word impulses is a fairly old word, is that this is actually an action potential. But I'm going to give you very brief idea of how this works. So, since it's a lot of molecules and ions and stuff, we go into it in more detail in the next semester. So what happens in an axon is an imbalance in ions is created between the outside of the membrane and the inside of the cell. It's a potential, is that there's stuff that could happen there, but the membrane, once you get this set up with a whole bunch of positive ions, a whole bunch of negative ions, and an imbalance between the two sides, then as soon as you open literally the gates, they call them gates in the membrane, then everything's going to rush through because they're going to try to balance. That setup is your resting potential. What happens with an impulse is the gates open. So at the very beginning of the axon, the gates open, and you get this flood of ions through the membrane. The membrane depolarizes. It was set up with unequal charges, so it goes even, it depolarizes. And that kicks open the gates farther down. Depolarizes, kicks the gates open farther down. So an action potential, an impulse, actually travels down the axon at a given speed for a given axon. Every axon has a set speed that the impulse can travel down it as. I'll come back to that in a second. And then behind the impulse, the gates slam shut and the pump's set to work, setting the resting potential back up. That's your recovery stage where, okay, this has all gone nuts. Let's put it back the way it was. So the impulse is traveling. Gates opening, well, if we're going this way. Gates opening here. This is the flood of ions. This is the impulse. And gates closing and reset behind it. This is why insects need fibrillar muscle, because the impulses do need to be reset. You can only send so many impulses in a row down. There's going to be a little time gap between them. Fibrillar muscle can contract faster than the inflow of axons, inflow of impulses can keep up with it. The bigger the membrane, the bigger the axon, the faster this can go. There's more gates around the edge. That's what you find in invertebrates that don't have the myelin insulation. And in fact, some of the earliest research that was done on how neurons work were done with squids. Squids have these huge axons that connect one end of the squid to the other end of the squid, where speed is of the essence to get the message back and forth. And so they use squid giant axons to figure out all of the details of what ions are doing what. We have wrapping around our nerves. So the ions are kept very close to the membrane. They can't get very far away. They build up in that little bitty gap. So it's a very efficient impulse. You open the gates, poof, they're right there. If you watch an impulse go down, okay, here's a wrapping cell. Here's the next wrapping cell. Here's the next wrapping cell. Impulses on vertebrate neurons jump. They go, because where there's a gap between the cells, there's a slight slowing down of the impulse because you're no longer holding the ions really, really close to the membrane. So the impulse will jump down from one gap to the next gap, but it still goes pretty doggone fast. We've talked about synapses before. There are some animals where the synapse isn't really a synapse, is that the nerve membranes are directly connected to the nerve membrane of the next nerve. It's called a gap junction, but it is sometimes called a direct synapse, which is kind of like saying it's a gap that's not a gap. Typically, the synapse that you talk about is a little bitty gap. And you're talking about releasing neurotransmitters on one side and picking them up on the other side to start up a new impulse and perpetuate it. The receptors don't hang on to the neurotransmitters for very long, and there are enzymes in the synapse to break the neurotransmitters down, and then the first cell takes the pieces in and reconstructs neurotransmitters. So there's a lot of recycling. Release, attach, release, break down, recycle, absorb, recycle. Systems can be diffuse or be centralized or be some combination of those two. In echinoderms, pentaradial things like starfish, is that the nervous system, there is no central brain, but there do tend to be collections for each of the pentaradial parts, like the arms of a starfish. In something like an octopus, you've got a pretty decent central brain, but you also have some pretty major centralized systems for each arm, which not only coordinate the response of the arms, but also there's a lot of responses there also. They are like little mini brains. So information going back in from the arms doesn't necessarily go to the brain for decision making. It may go into the collection at the base of the arms for decision making. We talked before about cephalization. Concentration of processors and senses at the front end of bilateral animals. Why bilateral? Because with two sides driving you through the world, you tend to lead with a particular end. And knowing what's there before you run into it, probably a pretty good thing. So you put your receptors and your processors at the leading end of a bilateral animal. Radial animals, no set directionality, unless you're a sea cucumber, and then you have some cephalization in a sea cucumber. Collections of nerves are ganglia. When a ganglion gets big enough, then it's considered a brain. There's no magical threshold for that. It tends to be just what people are used to calling particular collections of nerves. Is that I have been outsmarted by animals that have ganglia that don't have brains. Unfortunately, I don't want to run a really, really long video, or I'd tell you the story about how, as a freshman, I was outsmarted by flatworms. It's an interesting story. I might actually put it on a separate video for people that are interested. The type of processing you see, here's a problem. It's really hard to talk about brain processing and not compare it to computer processing. And some of it does compare fairly easily. There's simple processing where the steps are very much step, 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 step. That's linear processing. There's some processing where a collection of information will come in and it will be split into different collections of processors that will then recombine at the end for a decision. That's nodal. Then there are processors where it goes a whole bunch of different areas and it may not all be recollected or not all recollected kind of in a linear fashion, where there's a lot of processing going on parallel at the same time, multi-parallel. This is the advanced computer systems that we're dealing with now, do a lot of multi-parallel processing. You get into the issue with the issue of what the hell does this mean? Generally, we're talking about processing that we can compare to human processing, problem solving, certain patterns of responses to the world around us. And so we want intelligence that matches our intelligence. Is that the only type of intelligence there is? It depends on who you ask. But in terms of being able to solve problems that we kind of give them to solve, There's intelligence in the invertebrates. The cephalopods turn out to be extremely intelligent. Both the octopus and the squids and even the cuttlefish are pretty intelligent animals. They will respond. And if you go online and look for octopus intelligence, there's a huge number of videos showing them being able to do different things. And in one case, I was watching one fairly recently where they solved a problem in a way that the people that put the problem together didn't know they were going to be able to solve it. So it's like, oh, that happens. That happened with me and the flatworms, where they solved a problem in a way I had not anticipated them being able to do it. And then on the fly doing an experiment, you're like, oh crap, what do I do now? And I made the wrong decision. Anyway. Intelligence gets weirder when you get into arthropods, things like hive mentality for insects, where there's a lot of computer programming that's looking at this. An individual ant is not a terribly smart animal, but a collection of ants in a lot of ways acts like a collection of brain cells, where they interact and collectively are able to solve problems and do things that we would never expect an individual ant to be able to do. And so is that intelligence? It kind of, we'll talk about this in behavior. So it's a very, very tricky thing. And just trying to come up with a way to... If you look at the studies, it's always very interesting to see how people decide how to test intelligence in an animal that's not people. And they often are very good at giving them solvable problems within their range of responsive abilities. And sometimes you look at them and go, really? That doesn't really make any sense. As I don't know, what you're getting out of them just doesn't really fit what they're capable of doing physically, or the way that they see the world. Is that somebody did a paper on their first paper where they were showing horses pictures of things. And I'm watching this going, so do they even know that the horses see on the screen images that we see on a screen? And so far as I could tell, no, they hadn't done that. And if you have dogs or cats, you know that what they see on a TV screen does not seem to be exactly what we see on the TV screen. They see moving stuff, but they don't necessarily see another dog on a screen if there's a dog up there. If there's a sound, they respond, and then they start to see the motion. And what are they seeing? We don't really know. Anyway, that's a good place to stop.